The overall goal of this experiment is to visualize the microstructure of and cell adhesion on a non-transparent knitted titanium nucleus implant using fluorescent imaging techniques.
This method advances the analysis of tissue engineering with non-transparent scaffolds. One advantage of this technique is that one can trace the cell viability as well as the proliferation on the scaffold with defined culture period.
Visual demonstration of this method is critical, as the fluorescent staining and the later visualization is challenging. Individual scaffold properties, such as the pore size, may affect the timing, and autofluorescence of the scaffold may affect the choice of fluorophores that you are using.
Place up to 5 scaffolds, 6 or 7 millimeters thick, into a 50 milliliter tube and wash them with water 3 times for 20 minutes per wash. Do this at room temperature with gentle agitation. Next, wash the scaffolds in 30 milliliters of a 1% Triton X100 solution under the same conditions. Then perform 2 more water washes. Now sequentially sonicate the scaffolds in reagent-grade 99% acetone, 99% isopropanol, and 99% ethanol. Perform each sonication step twice for 5 minutes per sonication. Then sonicate the scaffold 3 more times in water for a total of 15 minutes. Finish up by placing the scaffolds on a lint-free tissue and air drying them overnight at room temperature. The next day, autoclave the scaffolds for 15 minutes at 121 degrees Celsius and 15 psi. To confirm that the cleaning worked, use indirect fluorescence.